Today, I'm going to focus on one aspect of the viral replication cycle, and that's coronavirus entry. And I'm really going to try to give an overview of what we know about the coronavirus entry pathway, and then talk specifically about places where one might intervene in the pathway in order to try to block infection, and try to put that in the context of what I know is going on right now in various labs here to try and target this pathway. So if we start looking just at a model of the SARS virion, so the SARS coronavirus 2 virion, and for sake and simplicity's sake, from now on, I'm going to call this SARS-2. The virion is shown in cutout here, and the main feature on the surface of the virion are these very, very large prominent spikes. And in fact, the way it was given its name was because corona means crown, and a cryo-EM, or in an EM, transmission EM at the time, these things really looked like a crown of spikes on the surface. So this is the most prominent glycoprotein in the membrane, but there are other proteins in the membrane like the M protein and the heme gluten esterase protein. Now, this is an enveloped virus, and what that means is that the virus is surrounded by a lipid bilayer, and the genetic information sits inside a lipid bilayer and a proteinaceous capsid. Now, it's important that this is an enveloped virus because enveloped viruses have a barrier that they have to get across, and that is they have to take their lipid barrier, their lipid envelope, and fuse it with the host cell's lipid envelope in order to accomplish the process of entry into the cell. And this is an energetically very unfavorable reaction. And so all enveloped viruses contain on their surface these proteins, these spike proteins, often in this case called S1 and S2. For HIV, they're called GP120 and GP41. These proteins always consist of a globular head domain and then a more linear transmembrane domain. And the way these work is that this globular head domain you could think of as the governor that controls the activity of this membrane-anchored protein. And the membrane-anchored protein is actually the machine that will drive fusion. And as I'll go through in some detail through a series of conformational changes, viral glycoproteins on the surface of viruses cause fusion, in this case, of the host membrane with the viral membrane. And in doing so, they allow the genetic information that's inside the virus here to be delivered into the cell through this pore that forms. Now, to accomplish this, the virus glycoproteins really have to undergo a lot of gymnastics. And so for coronaviruses, and for SARS in particular, we know that the virus exists, in a fusion incompetent state on the surface of the cell, on the surface of the virus. So when it's incorporated into the virus, it's fusion incompetent. It has to be primed, and that priming event is similar to the priming event that occurs for a number of related, and by related, I mean structurally related viral glycoproteins. So there's a whole class of viral glycoproteins called class 1 viral glycoproteins. And these viral glycoproteins all have this very similar globular head stalk kind of structure. They all have to be primed by a protease priming event that makes them now fusion competent. But as you might guess, if you're fusion competent and you undergo this molecular gymnastics at the wrong time, then you're going to waste this viral glycoprotein because it won't be in close proximity to a target membrane, a host membrane that it needs. Boy, I keep jumping. So all these glycoproteins require a trigger event. And that trigger event takes this fusion competent form into a form in which the protein has changed conformation and inserted part of itself into the target membrane. And the part that it inserts is called the fusion peptide. Then again, through some more gymnastics and more refolding of the protein, these proteins actually bend back on themselves, and in doing so, do work on both the host membrane and their own viral membrane. That allows these membranes to become very closely opposed and start mixing. And then there's resolution of what we call this hemifusion intermediate to form a fusion pore. and this final structure here, where essentially both ends of this protein that were in opposite membranes now are in the same membrane together. Now, how viruses accomplish this, we understand a lot from a number of studies over the years on especially initially the influenza as a model system to study this, and then HIV, and we understand a lot about how the HIV glycoprotein works, and SARS coronavirus. And these are sort of triggered, but how it gets triggered is very different. So for example, for flu, there's this priming event where it's cleaved by extracellular proteases. So now it's in a fusion-competent form. It then binds to carbohydrates on the surface of the cell, sialic acid. It's endocytosed, and the low pH environment of a late endosome serves as a trigger to allow the virus to do these molecular gymnastics. HIV is a bit different. So HIV uses as its trigger binding to two proteins, CD4 and then a co-receptor. And upon binding to the co-receptor, again, the virus undergoes, the viral glycoproteins undergo these conformational changes that allow fusion peptide insertion and eventually membrane fusion. SARS coronaviruses are different. And what our lab showed for SARS-CoV was that what happens is that SARS-CoV binds to its receptor ACE2, but that's not sufficient for triggering the rearrangement. It then goes into the cell, into an endosome, is what we showed, where it's cleaved by a protease. And protease cleavage within this endosome is actually required in order to allow the virus now to undergo its rearrangement. So it's actually cathepsin cleavage in this low pH environment that allows the conformational changes to occur, fusion peptide insertion and so forth. Now, it turns out that SARS is a little bit different than other viral glycoproteins that many of you probably know, in that SARS has two cleavage events. It has that first priming event that's very similar to the same kind of priming event that occurs for the HIV envelope, where it's cleaved from GP160 into GP120 and GP41, or for influenza A, where it's cleaved into HA1 and HA2. In this case, the cleavage event to generate S1 and S2 primes it, but it's not sufficient for the protein to be completely fusion competent. And the triggering event is actually cleavage at this site here called the S2 prime cleavage site. The S2 prime cleavage site is relatively close to the fusion peptide and allows the fusion peptide then to be able to free itself from this structure and to insert into the target membrane. That's another sort of unique feature of the coronaviruses, is that their fusion peptide is not near the amino terminus after the priming event. So the priming event for most class 1 viral fusion proteins puts the fusion peptide at or very near the amino terminus of the membrane spanning protein. So in the case of HIV, that would be GP41 or HA2. In the case of S2, the fusion peptide after the priming cleavage is not near the amino terminus. It's only after cleavage at the S2 site that the fusion peptide is actually near the amino terminus of the S2 subunit. And another sort of a unique feature of SARS-2 versus SARS is this, as Susan mentioned, is this acquisition of a furin cleavage site. And so furin likes the sequence RRKR or RRXR. And you can see that here in SARS-2, there's been a 4 amino acid insertion relative to all these other SARS-like coronaviruses. And you can see that now there's a RRXR motif, a perfect furin cleavage site. And if you look at the processing of these proteins in comparison to one another, and this is from Stefan Pohlmann's recent Cell paper, if you look at SARS-S, you can see that almost all the protein that's out on the virion is not even primed. It's still in the full-length SARS or S configuration. Whereas if you look at SARS-2, there's almost complete cleavage of SARS-2 spike down to S1 shown here, and S2, which isn't visualized by this antibody. So you can see that these viruses are very different. And as Susan mentioned, a number of groups have noted this, and they've compared it to influenza. So we know for highly pathogenic avian influenzas, for example, they acquire a much better cleavage site between HA1 and HA2. And it's been hypothesized that this much better cleavage site between S1 and S2 is part of the reason for the high pathogenicity of this virus. But there really is no evidence to support that conclusion yet. One study out so far has shown some modest degree of difference in infectivity of SARS-2 with a mutant cleavage site versus SARS-2 with an intact virion cleavage site. But the difference was not great. And so it's still, I think, and until we put these into an animal model, a robust animal model that recapitulates disease, we really don't know whether this virion site contributes to the pathogenesis or even the transmissibility of the virus. So I mentioned that we had discovered back on SARS-1 that SARS can use cathepsin L within the endosome, shown here, to do that activating cleavage. That second S2 cleavage can be accomplished by cathepsin L in the endosome in certain target cells. Well, it turns out, and that leads to the conformational changes, membrane fusion, and infection of the cell. It turns out that, and this has to, I forgot to mention, this has to occur after binding to ACE2. So Graham Simmons, when he was in my lab, did a wonderful job showing that if you used soluble ACE2, or if he did this trick where he used virions that would fuse to one another based on having receptor in one virion and glycoprotein in the other, through a bunch of really clever tricks, he was able to show that you had to bind ACE2 before you did that S2 cleavage. If you did it the other way around, if you cleaved with an enzyme that would cleave at S2 and then bound ACE2, you actually did not generate highly infectious virus. So subsequent to our analysis of cathepsins, it was noted that in the airway there are a number of other proteases. This was known for a long time. And this one protease, TMPRSS2, had been found to be very important for cleavage and processing of influenza glycoproteins. And so it was looked at, and it was discovered that TMPRSS2 can also do the processing at the S2 site, and in fact does it very efficiently. And if you have TMP, and this is a cell surface enzyme in the airway epithelia, if you have surface-expressed TMPRSS2 along with ACE2, you can mediate very efficient entry at the surface of SARS. And so it looks like, and this isn't true just for SARS, we think this is probably true for a number of coronaviruses, that they can use multiple different pathways to enter the host cell. Susan Weiss's lab has shown this for a number of different MHVs, that you can go into the cell in different ways, depending upon the particular MHV and the particular cell type. So really, dependent on trafficking, there's going to be very different requirements for cellular proteases. And so when we start to think about trying to inhibit infection by this virus and targeting different proteases, we have to be cognizant of the fact that numerous different proteases can carry out this S2 processing event. And really, as shown in the next slide, it's dependent. The results you see are dependent on the cells that you use. And so this is, again, some data from Stefan's recent paper. And what he shows here is that if you take viro cells, now viro cells don't express TMPRSS2. If he makes them express TMPRSS2 or looks at the control, in the control cells, if you use an inhibitor of cathepsins, E64D, you can see very efficient inhibition of infection by SARS and SARS-2. However, if you use canostat, which is an inhibitor, it's actually a drug, an approved inhibitor of TMPRSS2, TMPRSS2, you can see that there's really no effect on infection when you look at regular viros. However, if you overexpress TMPRSS2, you can see a modest effect of canostat, And then if you do camostat and E64D in combination, you can see that both SARS and SARS-2 are pretty effectively inhibited by those two drugs, or those two inhibitors. If you look at an airway epithelial cell, so Calu-3s are an airway epithelial cell, what you can see in Calu-3 cells is that if you look at MERS, SARS, or SARS-2, you can see that these guys are all inhibited in these cells by camostat. So camostat on an airway epithelial cell line looks like it's a very good inhibitor. Again, I think until we actually look in animal models, we really won't know whether camostat is an effective inhibitor. And my gut feeling is that developing protease inhibitors that target the cleavages of the glycoprotein, not protease inhibitors that target the virus's own proteases, but ones that target the proteases that are processing the viral glycoproteins, is going to be very difficult, because I think there's a lot of redundancy in the enzymes that can carry this out. And so I think that's going to prove to be pretty difficult. Interestingly, it was also found that SARS requires a calcium-rich environment, and this didn't make sense for a while. But if you can modulate the calcium levels, you can show that you can block either TMPRSS2-mediated fusion at the plasma membrane, or you can block fusion in the late endosome that's mediated by cathepsinel. And it became apparent why this was, why we saw these results, or why the field saw these results, when the fusion peptide was studied a little more carefully. And the fusion peptide of SARS coronaviruses, both SARS and SARS-2, they are a little bit different than other class 1 viral fusion peptides, and they just don't look quite the same. And so using some peptides and studying them, it became clear that these fusion peptides actually require coordinating calciums to hold them in the active conformation. And so if these peptides aren't in this coordinated conformation, if you chelate away calcium, then they don't insert into the membrane properly in order to start that the whole gymnastics that the virus is going to do, where it inserts its fusion peptide in and then starts undergoing conformational changes, dragging the host membrane closer to the viral membrane. So this may be another area where we can think about trying to attack the virus in trying to modulate in intracellular and extracellular calcium levels. But again, I think that might be a little bit difficult to accomplish. Now, it's been amazing in the last month to see the number of structural analyses that have come out already on the viral glycoproteins, and we've learned some really interesting things about the viral glycoproteins, where it's these viral glycoproteins again they're the same class as HIV and flu and other class 1 viral glycoproteins, but they seem to be doing things a little bit differently. And so it turns out that by cryo-EM, a number of different structures were seen. So this is the spike. This is a soluble form of the spike that is imaged by cryo-EM. And you can see that in this paper, they saw 5 different conformers of the spike. And starting on the left, this would be a conformer that would be not bound to ACE2 and in what we would call a closed conformation or a down conformation. This conformer has one of the 3 subunits. You can't really see because they didn't color in this cryo-EM, but there are 3 identical subunits here. That all these proteins work as trimers, which I forgot to mention to you guys, but in this trimeric structure, in this conformation, one of the conformers has opened its head and has opened up, and this region here is no longer tucked away and it's open. And in fact, it's this open conformation that then can bind to ACE2, and you can see that you can have a conformation, you can see, I keep pointing to the wrong screen. You can see that this conformation here now is opened up even more and has bound to ACE2. And then there are 2 other conformations that are ACE2 bound conformation. And it's unclear, I think, right now whether all three of these are normal conformations, whether they are just seen by cryo-EM, or if it really indicates that this is moving around here and that there's lots of flexibility. It could be, and the data suggests because of this process here, that this head of this glycoprotein is very mobile, and that these three head groups are moving around quite a lot. And that's important if we start to think about development of good antibody responses, because if these guys are moving around and in a number of different conformations, it will be difficult potentially to develop effective neutralizing antibodies that see all of these different conformations. So, for example, if all three of these conformations are competent to move forward towards membrane fusion, then it might be difficult to target all three. This glycoprotein seems to be exist in a closed conformation that then can open up, and it's when it opens up that it can bind receptor. Now, interestingly, in this paper, they didn't see molecules with more than one receptor occupied, and it's unclear whether you only have to occupy one receptor binding domain to start the triggering conformations required, or if you have to occupy more than one in order to begin triggering the conformational changes in this. And that will be important when we begin to think about developing antibodies that maybe bind to this conformation and stabilize it. You have to bind to all the sites in order to stabilize it, or can you bind to just one of the sites and stabilize the conformation? So based on these results and a lot of others over the years, a model has been developed for different stages of SARS Spike membrane fusion. And so what's clear is that there's an activating proteolysis that occurs. That proteolysis is shown here initially, but that proteolysis occurs after ACE2 activation, probably. So ACE2 interacts and binds. It won't bind, obviously, to the down conformations because the binding site is covered. So when the binding site becomes accessible, SARS-2 can bind. You have proteolysis to cleave at the S2 position. After S2 proteolysis, the molecule can start to undergo conformational changes. One of the very early things that we think happens is that the whole head domain is essentially shed off the molecule. And so you're left with just this portion of S2 cleaved at the S2 prime site embedded in the viral membrane. This then undergoes conformational change where the fusion peptide shown in red here then becomes exposed and inserts into the target host membrane. And then you have refolding of the viral glycoproteins to finally accomplish membrane fusion here. So refolding to bring the membranes together and then finally to fuse the membranes. So this is in many ways similar to what we know about all other class 1 viral proteins, but there are a number of different wrinkles involved in the process here. So if we start to look more broadly at not just SARS and SARS-2, but at all of the SARS-like beta coronaviruses and look at homology among them, it's kind of striking that this is a structure of SARS-2 with the regions that are similar across all of the SARS-like coronaviruses. These are all the viruses that have been seen in bats and people. And what you can see is that there's a very conserved stalk domain where the fusion machinery is, and a much more highly variable S1 top of the molecule. So the receptor binding domain would be, the receptor binding domain would be right here. We have this highly conserved stalk. The fusion peptide is buried back in here. And so the fusion machine is highly conserved. The regulator of that fusion, the thing that binds the receptor and starts the whole process, is much more variable. I think that's interesting in that it suggests, although there is no evidence to support this yet, that there could be antibodies or stabilizing molecules that could affect more than one of these coronaviruses. So you could potentially have target the stalk to develop antibodies or therapeutics that would be more broadly reactive. Whereas if you start making antibodies up here that they're going to be much more specific for the SARS or SARS-2 or the other coronaviruses. And that's supported somewhat by the data. We know that there are mouse polyclonal sera against SARS that can cross-neutralize SARS-2. We know that there are human polyclonal sera against SARS that can cross-neutralize SARS-2. In a study of 4 different monoclonal antibodies that all targeted the SARS receptor binding domain, 4 of 5 did not bind to the SARS-2 receptor binding domain and did not neutralize SARS-2. So I think the jury's still out as to whether antibodies against SARS are going to be effective against SARS-2. Clearly, polyclonal sera will have some reactivity, but we don't know whether that reactivity is against the head domain or against the stalk. And I think that's going to be a very interesting area of research over the next several years. I mean, people who work on flu know that the flu field is talking about developing a universal flu vaccine that's very focused on the stalk region. And it's unclear whether there are good antibody epitopes in the stalk domain, or whether for some reason that region is not effectively immunogenic in the mouse models. If we just took a sort of, and I don't expect anybody to be able to read this, all I want you to look at is black versus not black. And so the top 4 lines are, 5 lines are all different SARS-1 viruses or SARS-like viruses. And then the bottom line here is SARS-2. The second line up is SARS-2. And what you can see is that this is the beginning of S1, and this is about 3/4 of the way into S1. Halfway into S1. The receptor binding domain is in here, in this region here. And you can see that although there are blocks of high conservation between, say, for example, this region here, between SARS-1 and SARS-2, there are clearly regions that are significantly different. And there's an insertion in SARS-2 relative to SARS-1. So these regions, when you look at them overall, they are similar but clearly not identical. And that's very different when you look at SARS-2, SARS-1 versus SARS-2 in the S2 subunit. So again, the second line from the bottom here is the S2 subunit. This is the membrane spanning domain right in here. You can see that they're almost completely conserved SARS-1 to SARS-2 with a few amino acid changes scattered throughout. But SARS-2 and SARS-1, the S2 subunit, is very highly conserved. So again, one might imagine that if you could use these as an immunogen, you could elicit antibodies that will see that stalk and potentially neutralize. Again, we don't know yet whether there are going to be good antibodies that can target this, but I'm sure that when people start looking at the human antibody repertoire in these guys and cloning human monoclonals, we'll have a good idea in the next few months whether we can target stalk. And then the other question that comes up a lot is how much sequence variation is there in SARS-2 spike domain. So now these are all just different SARS-2s from around the world, China, Europe, the United States. And what you can see, again, don't look for anything but black versus white. You can see that in this region of over 700 amino acids, 740 amino acids, there are 3 changes that have been noted, and most of those changes are in one virus or another and are probably sporadic in nature and do not appear to be coming fixed, meaning that they're giving any sort of advantage to growth in humans or in human cells, or that they're escape mutations, although at this point I don't know that we'd be expecting antibody escape mutants to become fixed in the population because there's really no pressure in the next individual who gets infected. So the receptor binding domains of these guys are highly related, but clearly divergent from one another. And so the question initially was, do they confer the same tropism? So are these viruses, SARS and SARS-2, similar in terms of the kinds of cells that they can infect, and are they similar in the kinds of species that they can affect? And so what you see here is, this is from, again, from Stefan Pohlmann's recent paper. What you can see here is a number of different human cell lines. Oh, come on. A number of different human cell lines. Some are infectable. Some, like MRC5s, are very poorly infectable. Very little ACE2 on MRC5s. Interestingly, if you look at bat cells shown here, a couple of different bat cell lines, so bats are here, so bat cell lines, you can see that the bat cells, although they're down a little bit for VSV, a couple logs down for VSV itself as a control here, they're really not very susceptible at all to SARS or SARS-2. So this virus has clearly evolved when it left bats, or these particular bat cell lines are not representative of the bats that SARS came from, the ACE2 that's in these guys. So we still don't know that for sure, but it looks like this virus has adapted into people quite well and can infect people. These are viros, which are African green monkeys. Those are also infectable. Interestingly, I think these are MDCKs. Somebody talked about dogs before. These are canine kidney cells, and you can see that MDCKs are pretty infectable. But again, the data out there as to whether pets can be infected is all over the place, so I don't think we know. And as was the case for SARS-CoV-2 also does not use the mouse ACE2 receptor apparently very effectively at all. And so again, animal models for SARS, small animal models, are going to be engineered by putting ACE2 into these animals and doing knock-ins. And so breeding these mice up and getting enough mice for a lot of people to work on them is going to take a little while. So I just want to talk a little bit about how we're trying to begin analyzing these viruses, and then talk more about what's going on at Penn. So my lab and a number of labs around the world, including Stefan, some of the data that I showed, we use pseudotypes to look at how these viral glycoproteins work, how antibodies to them neutralize, because effectively when the virus has the glycoproteins on its surface, whether it's SARS itself or whether it's pseudotype, from everything we know, not just from SARS, but from pseudotypes for a large number of different viral glycoproteins, the glycoproteins behave like they're on their own virus when they're on the pseudotype. So what is a pseudotype? Well, a pseudotype literally is just a virus that's a Trojan horse. It's a virus that has somebody else's glycoproteins on the outside, so it looks like that other virus. The way we make pseudotypes now is that we use a VSV, the vesicular stomatitis virus, that doesn't encode a glycoprotein, but does encode a green fluorescent protein. We introduce that into cells that are expressing high levels of SARS-2 spike. We then harvest off the pseudovirions from those cells now, so they have these red spikes on them derived from the SARS-2 plasmid that we put into these cells. And then we use these pseudovirions to express ACE2 or ACE2 expressing or ACE2 negative cell lines as controls and analyze reporter gene expression. The reason we like this assay is because we can generate large amounts of this virus stock, and we can do this on a per cell basis, and so we can read out titers and neutralization and everything as a focus reduction neutralization titer assay, or FRNT. So we can do this relatively high throughput because it's all in 96-well microneutralization format. We also generate recombinant VSVs, and recombinant VSVs we're in the process of generating for SARS-2. Recombinant VSVs have a couple of advantages over the transiently produced pseudotype VSVs. They still are safe. You can use them in a BSL-2 environment. It replicates very quickly. We built fluorescent reporters into this guy. The nice thing is you can have a common backbone for different viruses, and then you can focus on what's happening to that particular virus glycoprotein with good controls. So for example, in this case, we'd be using VSVG as a control, but we've built viruses that have the Ebola glycoprotein, hantaviral glycoprotein. So you have a number of controls here. It's really scalable for high throughput screening. So once you make these recombinant viruses, they grow, and you can produce, lots and lots of particles. And in fact, you can use these particles for more than just infection. You can use them to pan for antibodies or B cells that recognize epitopes that are displayed on the surface of these particles. They can also be used to select for neutralizing antibodies and inhibitor escape mutants. So for example, let's say you develop a monoclonal that neutralizes SARS-2, and you want to find out what happens. Well, rather than having to grow SARS-2 in the presence of monoclonal and look for escape mutants, we can do that in these viruses, and we can look for the glycoprotein to change and escape that monoclonal or escape that inhibitor that's inhibiting viral entry. So it happens very rapidly. The good thing is unlike coronaviruses themselves, the mRNA, the RNA-dependent RNA polymerase of these viruses does not have a proofreading activity, and so it's much more error-prone, and so you can generate mutants much more rapidly. And then the final thing is that you can also use this as a vaccine platform, either alone or in conjunction with other modalities. And in fact, it's this virus here without M. cherry that with the Ebola glycoprotein inserted into it, that is the Ebola virus vaccine that's being used in Africa and has been now in hundreds of thousands of people. So we know it's a safe platform for immunizing people. VSV itself generates, or VSV and VSV Ebola, they induce very good both humoral and cellular responses. So it's probably a great platform to look at generation of both cellular and humoral responses. So I want to talk very quickly in the last few minutes about interventions that one might do to target SARS-2 entry. So, for example, one might think about generating antibodies that would block the receptor binding domain, either monoclonals or vaccine-generated antibodies that could inhibit this. Or you could use convalescent sera that might inhibit receptor binding. And so these are being developed on campus by a number of people, vaccines by Norbert Party, Dave Weiner, Henry Lee's working with a group and us on some human monoclonals. Bihan and Katie are working on getting convalescent sera along with Ian Frank. And Scott is doing a lot of analysis looking at what people have good ELISA titers to then look at in these kind of experiments. You can also look at potentially focus on stalk. And again, this hasn't really been done yet, and it might be a good target to look at because, again, it's highly conserved across the SARS-like viruses and could present a target that gives you a good broadly neutralizing antibody or BNAB. As I mentioned, these viruses all require a second protease activating cleavage, and so the S2 prime proteases, you could go after those. You could go after TMPRSS2, and in fact, there is a licensed drug that inhibits TMPRSS2, cathepsins, etc. And Sarah's screening for inhibitors of MERS. Some of those almost surely will be protease inhibitors that are likely targeting cellular proteases. As I mentioned, one problem in doing this is that the proteases may be redundant, and so what you see in one cell line when you do your screening may not pertain to what's happening in the lung or in other cell lines. So you have to be careful when you do those experiments. Small molecules, peptides, proteins that block ACE2 interactions. Avery Posey has a number of lectins and other things that will bind probably to the carbohydrates all over here and could potentially block this interaction. Norb Pardee is making an ACE2 FC fusions that will be expressed and will be able to essentially decoy the virus and bind to the ACE2 binding site and block infection. And then there are some other ideas that are out there. So small molecule entry inhibitors. So a number of labs over the last several years, and this is a paper from PNAS just in March 2018, a number of labs are starting to develop small molecules that actually fit into the structures and they stabilize the viral glycoprotein in its prefusogenic structure. So it can't undergo the conformational changes it needs to undergo to become fusogenic. And so you could imagine that those could be very effective inhibitors. And so those would block conformational changes, either the opening of the head or potentially the refolding that's going to be required to release S1 and get S2 into its fusion conformation, or even to do the fold back. And we actually know of a drug that inhibits this. So the HIV inhibitor, it's a small peptide inhibitor, infirmitide, is actually a peptide that binds here and inhibits this fold back reaction. And so we know that these kinds of things are possible. They've been developed for other viruses, a number of other viruses, and so they likely will also be targets for intervention in the pathway. And then finally, there are some other things that you can do. So Cesar de la Fuente here is very good at designing membrane-active peptides, and he's collaborating with us. We're going to screen a bunch of peptides that he's designed that insert into membrane, and the idea is that they'll insert into the viral membrane and disrupt it in such a way that it can't go through this bend here, that it has to bend very tightly in order to allow membrane fusion to occur. And then Avery Posey has a number of lectins and other molecules that likely will be able to affect either conformational changes or binding to the receptor. So in summary, we know now that SARS-1 and SARS-2 appear to have very similar entry pathways, and they appear to utilize common host factors, although they do it slightly differently. The sequence of SARS-2 displays significant divergence, especially within the S1 subunit from SARS. And if we look at the family as a whole, all the beta coronaviruses seem to display significant divergence when you look at the top, especially of the S1 subunit. S2 has a unique priming site, and whether this furin priming site contributes to a spread and or pathogenesis remains to be analyzed. And then, as I talked about at the end, there's a lot of different ways and a lot of different targets in the entry pathway that we could look at for developing interventions. And then just to acknowledge the people in my lab right now who are developing all the entry assays, Phil Hicks, Thomas Mazzani, and Brian Ropers working with us, and then previous members of the lab, and then collaborators here, especially Stefan, who sent us a bunch of constructs already that we're just trying out. Thanks.